Welcome back to the playlist on amino acid catabolism. What we're going to do in this video is we're going to talk about the two amino acids that get degraded to this metabolite right here, and that's glutamate gamma semialdehyde. Okay? In a separate video, we'll actually look at the catabolism from there, but at least I want to converge to this product. Okay? And the two amino acids that get degraded to glutamate gamma semialdehyde are arginine and proline. And in the first half of this video, I want to talk about the easier one, and that's going to be arginine. And to an extent, if you've watched the urea cycle video, you're already familiar with the first enzyme, and that's arginase, right? That was part of the urea cycle. And at that point, we had synthesized arginine, right? We already saw the biosynthesis of arginine in that video. And if you recall, we took argininosuccinate and we reacted it with argininosuccinate lyase, and that cleaved off fumarate, right, which went into the TCA cycle, and that left us with arginine, right? And then we took arginase, which was a hydrolytic enzyme, and we got out urea, okay? And in the process, what we did is we generated ornithine. Now, one thing that you need to be aware of is ornithine really has two main paths. The path we saw in the urea cycles was where the ornithine got transported back into the mitochondria, right? And it reacted with activated ammonia in the form of carbamoyl phosphate, right? And that was done by the enzyme ornithine transcarbamylase, right? And that gave you citrulline, which got transported back out into the cytosol. So ornithine can go back into the mitochondria where it will react with ornithine transcarbamylase, or it can react with a transaminase. And the specific name of the transaminase is shown, if I can find my mouse, it's shown right here, and that's ornithine delta aminotransferase. Now notice what's happening. It's the same reaction, right? You're starting with alpha ketoglutarate and you're going to glutamate. So what we should have is we should have an amine right here, and you should expect that we should replace that with a carbonyl bond. Now if you remember the video where I talked about transaminases, if you remember, I said we're essentially replacing an amine with a carbonyl, and then the other molecule, we're replacing the carbonyl with an amine, right? It pains me to say this, but you can effectively think of it as a substitution between a carbonyl and an amine, right? That's at least the way you can conceptualize it on an exam, if they're not asking for the paradoxal phosphate-dependent mechanism. Now, if you remember from that video where we talked about transaminases, I said that 99% of the time when you do a transaminase reaction, the carbonyl that you generate is a ketone, right? This is that other 1% of the time. Notice that it's not the alpha amine that we're doing the transamination with, it's actually the delta amine on ornithine. Right? So it's this amine down here on the R group. Remember, ornithine's an amino acid, right? It's an amino acid. It's a strange one that you won't find in proteins, but it's an amino acid nonetheless, except we're doing the transamination with the delta amine. Okay? And so as a result of the carbon that's part of the amine, as a result of it only having one other carbon group attached to it, what we're going to end up generating is an aldehyde. So this is the aldehyde we're talking about, and that's what's characteristic of glutamate gamma semialdehyde. Okay, so that's the degradation of arginine, and honestly, it's a lot easier of a topic than proline. Proline's really interesting, and proline's actually a really fun topic. And before we go into proline's details, what I want to mention is that I'm going to be talking a little bit about apoptosis, and I'm going to assume that you've had at least a little bit on apoptosis, and I'll try to fill in a little bit of the details if you haven't. Apoptosis is programmed cell death. So there comes a time in which, let's say you have DNA damage to a particular cell. Now, ordinarily, if there's just a little bit of DNA damage, and there comes to be from time to time, if there's just a little bit, the cell has a pretty easy time repairing it. There are different kinds of DNA repair enzymes that repair the DNA, and then once you repair the DNA, you can go into the S phase of the cell cycle where you synthesize and replicate the DNA, and then you undergo mitosis, right? But the key is you have to have good DNA. In other words, you have to have mutation-free DNA in order to complete the S phase, right, before you can go into mitosis. Okay, but there comes sometimes when there's so much DNA damage that the cell, rather than wasting energy on repairing that magnitude of DNA damage, it instead undergoes apoptosis, and the cell just kind of throws its hands up and says, okay, for the greater good of this organism, I'm just going to kill myself so that I don't replicate myself. In other words, I don't divide and basically clone myself, which is what mitosis is. So instead of spreading the damaged DNA to future generations of cells, it just kills itself. And that's what apoptosis essentially is. And there are various mechanisms that take place during apoptosis. For instance, we might have a video someday on it, and you'd find that there's a whole bunch of caspases, which are apoptotic-inducing enzymes that get activated. And one other thing that you need to be aware of is there's a particular protein that is in very high concentration whenever you're undergoing apoptosis. And that's this protein right here, this protein called P53. P53, when it gets overstimulated, it induces apoptosis. And not only does P53 not only is it synthesized in high amounts when you're undergoing apoptosis, but you also get a lot of reactive oxidative species. And one of the enzymes that's able to synthesize reactive oxidative species is Prolene Oxidase. And the reaction that's shown here for Prolene Oxidase is not exactly correct, and we'll go into why that is in just a few minutes. Okay, but what I at least want to say, first of all, is there's a general cycle for Prolene Oxidase. And actually, a better name for this enzyme is Prolene Dehydrogenase. And actually, if you find the name Prolene Dehydrogenase in the literature, they're essentially talking about Prolene Oxidase. The reason some texts call it Prolene Oxidase is that one of the terminal electron acceptors in the mechanism can be oxygen, but it doesn't have to be. Okay, now what you must understand is we have proline, right? We have proline, and what we can essentially do is we can dehydrogenate it into something called delta 1 proline 5 carboxylate. And that product is what you see right here. It's delta 1, let me find my mouse, delta 1 pyrroline, excuse me, not proline, delta 1 pyrroline 5 carboxylate. Okay, so that's the immediate product of dehydrogenation. And specifically the electrons that are abstracted when we form the Schiff base, they go to FAD. Right? So the immediate product, at least from the enzyme's perspective, besides delta 1 pyrroline 5 carboxylate, is FADH2. Okay? Is FADH2. Now, those electrons can go to multiple sources. Okay? One of the ways in which proline oxidase functions is it can deliver those electrons from FADH2 to ubiquinone. Okay, so that would be coenzyme Q in the oxidized state. And then coenzyme Q can pick up the 2 electrons from FADH2, and that would generate ubiquinol, the reduced form of coenzyme Q, right? But there's another way that this can work. And what ends up happening is if you have molecular oxygen, right? And this is what they're talking about in this reaction where you have 1/2 oxygen molecule, right? You can have molecular oxygen, right? Let me do it like this. You have 1/2 oxygen, right? So they're just talking about 1 atom of oxygen. And it can pick up the 2 electrons, so it's a 2 electron transfer. It can pick up the 2 electrons from FADH2, and that's going to give you water. And of course, in the process, you'd have to also pick up 2 protons, right? So the key is, in any case, whether you're going from FAD to FADH2, or coenzyme Q oxidized to coenzyme Q reduced, or half of the oxygen molecule to water, it's always going to be a 2 electron, 2 proton process. Okay? But specifically what we're concerned with is the 2 electrons. Now, in all proline oxidases or proline dehydrogenases, the initial electron acceptor from proline in the generation of delta 1 pyrroline 5 carboxylate is FAD. And so what you're going to get is FADH2. Okay? Now, what changes in this enzyme is the terminal electron acceptor. And that is going to be, in one case, coenzyme Q, and you can imagine that it's going to be reduced into ubiquinol. right? And then the ubiquinol will go and then fuel into the electron transport chain. And specifically, it's going to feed into ubiquinol cytochrome C oxidoreductase, which is complex 3 of the mitochondrial respiratory chain, right? But one of the other electron acceptors can be molecular oxygen. Now, let's talk about why they put 1/2 oxygen. Okay, what they're trying to do in this reaction sequence, and quite frankly, they do it in most texts, where they put 1 proline goes to 1 delta 1 pyrroline 5 carboxylate, and that reduces 1/2 of molecular oxygen to water. The reason they do that is because they're trying to show 1 cycle of Prolene oxidase. But what it turns out is that in order to totally reduce 1 molecular oxygen, it actually takes 2 Prolene oxidase cycles. And keep in mind with this reaction that it doesn't matter what the final electron acceptor is. The FAD overall doesn't change in the reaction, because as soon as the electrons are accepted from FADH2, right, the FADH2 is going to be reoxidized back to FAD. So there's no net change in FAD, but the terminal electron acceptor is what changes, and that's what gets reduced. But a better way to view this reaction is like this. Instead of having a 1/2 as a coefficient, we can instead say, let's say we have 2 prolines, right? 2 prolines, and that's plus, we'll do it like this. We have 1 molecular oxygen, right? And by the way, this is going to be a 4 electron transfer process. In other words, we're going to have 2 cycles of Prolene oxidase, 2 cycles. And so what we're going to get is 2 delta 1 pyrroline 5 carboxylates. And then we're also going to get 2 molecules of water, so 2 H2O. That's a better way of putting this reaction. When they're saying that it reduces 1/2 of a molecular oxygen to water, what they're talking about is this is 1 cycle of Prolene oxidase. But in order to totally reduce a full molecular oxygen molecule, you have to have 2 cycles of Prolene oxidase, and you would end up getting 2 waters. Okay, there's 2 atoms of oxygen in molecular oxygen, and so you'd need 2 waters to account for that. So they're just trying to show you 1 Prolene oxidase cycle. Okay, and of course that goes to water. Okay, so I just want to drive that point home. And another thing that's interesting about this enzyme is that it's activated especially during the apoptotic process. Okay, and to understand that, let's talk about P53. P53 is a protein that's upregulated during times of DNA damage. And if there's a significant quantity of DNA damage, P53 gets overstimulated and it starts activating other proteins and enzymes. One of the enzymes that it activates is Prolene oxidase. So if the cell has already been committed to apoptosis by P53, Prolene oxidase starts getting upregulated, which means that there's an increased biosynthesis of Prolene oxidase. So what it means is that the highest amount of proline catabolism is going to be observed during the apoptotic process. Okay? And so you should expect that whenever you're undergoing apoptosis, you're going to have a lot of delta 1 pyrroline 5 carboxylate. And actually, one of the interesting things about this enzyme is that remember that the terminal electron acceptor can change. And so what can happen is instead of totally reducing molecular oxygen down to water, right? Instead of doing that, what can happen is you can take molecular oxygen, right? And keep in mind that the correct structure of molecular oxygen is like this, right? Essentially what would happen is molecular oxygen would undergo a homolytic bond cleavage, right? So the correct structure of oxygen is actually this, okay? And keep in mind that this form of molecular oxygen that's shown on the bottom, that's actually what the terminal electron acceptor would be in the form of. And let's say, for instance, that Prolene oxidase catalyzed an electron transfer into this molecular oxygen. Well, what you would generate is this. You would generate this. You would generate one of the oxygen atoms now has a negative charge. Okay, this is called superoxide. It's called superoxide, and it is termed a reactive oxidative species. And the reason it's called reactive is because one of the oxygen atoms, this one, has a radical electron on it, okay? And specifically, it's a very reactive radical electron. It will spontaneously oxidize proteins, and it will oxidize fatty acids and nucleic acids and all sorts of things that are bad for the cell. So part of the strategy in activating proline oxidase, or at least upregulating it during times of apoptosis, is that you're going to get an increased synthesis of reactive oxidative species. In fact, that's one of the proposed mechanisms as to why proline oxidase gets upregulated during apoptosis. In fact, when you're doing normal protein catabolism, proline is not one of the normal amino acids that's catabolized in high amounts. It's really catabolized, though, during apoptosis. Okay, so what I want to drive home, the main thing is that the way they've drawn the reaction is really stupid. Okay, is really stupid. They really should have said 2 prolines go to 2 delta-1 pyrroline 5 carboxylates, and 1 molecular oxygen is reduced to 2 water. So in all, if you were going to totally reduce molecular oxygen, it's going to be a 4-electron process. And if you've seen the electron transport chain videos, right, you've seen something like that. You've seen cytochrome C oxidase. That was an oxidase, and it took 4 electrons in order to totally reduce oxygen to water. It's the same type of thing. Okay, so let's go over the mechanism of that really quickly. So this molecule right here, this molecule is FAD. Remember that FAD is going to be the oxidized form of this particular flavin. And in the process of the 2-electron transfer of the proline oxidase cycle, FAD is going to be reduced into FADH2. So FADH2 is the reduced form, okay? And those electrons from FADH2 are then going to be shunted into ubiquinone, and in the process it'll reoxidize FADH2 back to FAD. So in the process of this electron transfer, we should get those electrons going into ubiquinone, which is one possible terminal electron acceptor. Remember, we can also have oxygen as an electron acceptor, but this is just one possible one. And in the process of picking up those electrons, ubiquinone gets reduced into ubiquinol. Okay? And remember that part of the structure of ubiquinone that gives away that it's at least it's oxidized, is going to be this quinone ring, right? This quinone ring is the giveaway that it's in the oxidized form, whereas notice that the quinol form is characteristic of the reduced form of coenzyme Q. Okay, and just remember that if oxygen is the electron acceptor, then it's going to take 2 cycles of proline oxidase to totally reduce oxygen to molecular oxygen to water. And I hope this makes sense, okay? 2 proline oxidase cycles are required in order to totally reduce molecular oxygen into water, and you should get 2 water molecules. Remember, molecular oxygen has 2 atoms of oxygen, and you would take 2 molecules of water to account for both oxygen atoms, okay? Now, one other thing that's really interesting about proline oxidase is it's actually thought that part of the mechanism of things like seasonal affective disorder and schizophrenia, it's thought that part of the mechanism is deficiency of proline oxidase. And you can certainly go into the literature and find theories about that. So it's just interesting to think about. Could potentially problems with apoptosis affect conditions like schizophrenia and depression and things like that. So just something to file away in the memory. And another thing that's interesting about this enzyme, we already mentioned that this particular enzyme is going to feed electrons into the electron transport chain, right? One of the electron acceptors, at least the terminal one, is ubiquinone, which gets reduced to ubiquinol. And if you remember, the ubiquinol is going to transfer its electrons into complex 3. Complex 3, which is ubiquinol cytochrome C oxidoreductase. So really, the ubiquinol is then going to transfer its electrons to cytochrome C. Well, that's interesting, right? Because if you remember, cytochrome C is part of the mechanism of apoptosis, right? It's part of the mechanism. In fact, cytochrome C is responsible for activating caspase 9. Okay, remember that caspases are enzymes that activate apoptosis, right? And so potentially Prolene oxidase, if it's able to feed its electrons into cytochrome C, it's going to change the oxidation state of cytochrome C, right, by channeling electrons into it through ubiquinol. So it's possible that prolene oxidase in large part controls the activity of cytochrome C and therefore has its hands in controlling apoptosis. Again, none of these things are really proven, but it's just things to think about, things that are kind of interesting. Okay, enough on that. At this point, we've generated delta-1 pyrroline 5-carboxylate. Now, there's going to be a spontaneous process that occurs in the direction of running from delta-1 pyrroline 5-carboxylate to glutamate gamma semialdehyde. And notice in this molecule that we've generated, right here in delta-1 pyrroline 5-carboxylate, there's a Schiff base, right? Essentially, when we oxidize proline, we get a Schiff base. And so what's going to happen is there's going to be a Schiff base hydrolysis. And if you need help predicting those products of Schiff base hydrolyses, we have a video on that. And so we're going to get a Schiff base hydrolysis. And what you would know is if you knew Schiff base hydrolyses, you would know that the products of it, in this case, is going to be an amine and an aldehyde. And that's exactly what we see in glutamate gamma semialdehyde, right? We have an aldehyde that's right here, right? And then we have our free amine that's up here. Okay, so that's enough for this video. We've seen that proline and arginine get degraded to glutamate gamma semialdehyde. In the next video, we're going to look at the fate of glutamate gamma semialdehyde. See you in the next video.